much higher albumin expression while using albumin secretion, sorry, while using the coating created from liver extracellular matrix gels compared to other regular hydrogels like collagen 1 or any other system. Additionally, to the almost quantitative adhesion of hepatocytes, which are very fragile, very demanding species to work with. So they never adhere quantitatively to collagen matrices. However, here, probably because of the complex 3D morphology of the layer, we managed to capture most of the cells on the layer and study them on the layer. So, sorry. Yep. But the most fascinating things start happening when you have fibroblasts. So fibroblasts form perfect self-assembled films or jello structures, even when you actually lower the concentration of extracellular matrices to subcritical point. So the point when it doesn't gelate anymore. So basically, we started seeing the self-assembly processes of extracellular matrices together with fibroblasts. And those assemblies are not static, though they are dynamic. They increase, they contract, and they release the strength upon external stimuli. And we can see that macroscopically, even in quite large objects, as 96 well plates. That's quite large objects for such microscopic events that are going there, which plays out in the whole macroscopic system, actually. So the cells remain viable within those layers, and we see that actual proliferation capacity and actual viability of those fibroblasts impacts severely the degree to which the extracellular matrix compacts. And we continue to study this system already in the industrial processes, just to improve on how do we control it, or what stimuli affected, or basically which more complex objects can we create of it, and what are their use in modern fibrotic diseases, or in cancer biology for this matter. So we thought further, okay, if we see self-assembly with fibroblasts, is there any other self-assembly with other cells, with more simple cells than fibroblasts in mechanical sense? Because fibroblasts, they express skeletal muscular actin, which from material scientist point of view is basically an elastic strength appearing, new elastic bands appearing in the system. But other cell types do not have so much skeletal muscular actin. Okay, will they produce a self-assembled structures? To some part, yes. So a lot of cells produce a self-assembled structure. Some of them are perfect gels, some of them less perfect gel. But yeah, we see the effect reproducibly in most of the cell types. And then we thought, okay, so if there is adhesion, which is very high because self-assembly is driven by adhesion for sure. So if there is adhesion, if there is contraction, let's combine them into one single system. So let's take the stromal cells, stromal component, which are again elastic. Let's take extracellular matrices, and let's take basically any other cell types and see how do they assemble. Do they assemble together? And that's where it's yes, they assemble together into the complex 3D systems, which can be obtained by quite simple laboratory procedures, unlike most of the things we know. Like spheroids we use, unlike organoids, which require weeks for establishment. You can do it in 24 hours in establishment of complex 3D systems. Okay, that's well enough. Let's go further. Can we make those objects smaller? They're quite large. So objects which is of the size of 9612, it's cool, it's visible, not very practical. Because if you want to do imaging, if you don't do complex experiments, you need to go lower in size. Lower in size. Let's combine those cells and add steering. Let's add steering into well plate and monitor what's happened. Not in well plate. Let's take a vial, take a magnetic stirrer, take a matrix, fibroblasts, and cells, and monitor what happened. So 15 minutes, we don't see much happening. We see binding of the cell. 1 hour, we see many more cells bound in the structure, and some form some morphology-less structures upon when we plate it after 1 hour of steering into low plates. 24 hours pass, look at the beauty of what happens. So classical remodeling and formation of more dense networks triggered by the stromal cells promotes the formation of organoids at the tempo which we never experienced before, which wasn't achievable before. Classical organoid culture is established within weeks and maybe not established at all. So success rate, we investigated these processes in the very early days of our research. So the success rate for those cultures is at the orders of 10%, 20% of the formation. Those systems can form assemblies at 90% of the success rate. Very high success rate, and practically any cell types that you want to use can be incorporated. So immune cells, meaning stromal cells, meaning cancer cells, meaning any kind of epithelial assemblies that you want to get. So the promise of those systems led us to any oncological, non-oncological investigations. We continue those, we continue expand, we continue now in the industry. So we partnered recently with several large pharma companies to develop more practical models of it, to bring it to the actual drug discovery processes. And that's what we will continue to do. And on this moment, I will thank you for your attention and wait for your questions.
Anton, thank you for your amazing lecture. Please, it's a time for your questions. Do you have any questions? May I start? I have 2 questions. There are a lot of situations requiring modern using ACM.
Yeah.
For example, it's most interesting for me. For example, immune cell homing during the disturbances with, for example, chemokines, during autoimmune diseases, etc. And second situation, very important is for me. For example, modeling specific tumor-associated ECM. Tumor-associated ECM is characterized higher stiffness as compared to the normal. Can you model this stiff AMC?
Yeah, actually, that's good question and quite a challenge. So actually, for us, the tumoral ECMs seem simpler to do than normal tissue ECM because of the stiffness. Because what we look for are the formulation for material creation, are the crosslinks, which is between proteins and glycosaccharides. And basically, the tumoral ECM is highly crosslinked. That's why it actually becomes very, very stiff. So actually, we work with, first of all, as a batch product with soft tissue sarcoma extracellular matrices. And now we signed quite amazing partnership with one of the pharma leaders in this field on CAR-T cell homing for in tumor microenvironment and basically in the context of colorectal cancer specifically. Mm-hmm. So we will continue working on that. And again, from our point of view, like the simplest are the stiffest. So the simplest tissues to convert to materials are the stiffest tissue there are. So we experience more problems with actually not that stiff tissues, like, for example, heart. Actually, heart is stiff because of the cardiomyocytes, because of acting in myosin networks. But those are cellular. Well, once you remove them, it's not that stiff anymore. So how, with that or those organs, we actually struggle more than with organs with high stiffness. And yeah, it's amazing chemical nature of the hydrogelling processes as well as actual extracellular matrix assemblies.
This is the second question. The next issue requiring the correct ECM is neurodegeneration, for example. In different brain area, we deal with the same, maybe the same ECM with tiny differences. Can you model these tiny differences in different brain areas? using this methodology?
Oh, yeah, I think so. It's unexplored. It's very, very cool to engage. And we right now moving towards more and more microscopic systems that we can create. So can we make the hydrogel from 1 gram? We can make it certainly from 200 grams. We can make it from large tissue. Can we make it from tissues are very small? To model not overall picture, but the local differences in those aspects. And I would say, if you will invite me within a year, I will already have answers to your question.
As maybe last question, can we use this ECM in the same way as Matrigel for example, study of tumor cell migration during the metastasizing?
Yeah, migration is being it's more complex in those gels indeed than with Matrigel, because Matrigel, again, the stiffness and the contraction plays a role. You can still use it for the migration of the spheroid experiments, but the wound migration will be a bit complex because of contractility. So it's basically moving outside than moving inside, which works better with system with this characteristic mechanics. So you can still model it, but you need a bit different approach. So like black and white.
Mm-hmm. I have no questions. Do you have any questions anymore? No. Thank you again for your outstanding question.
Thank you for your attention.